okay if we start at the back? So thank you everybody for coming. It's great to see so many people here. For those who don't know me, I'm Kristen Musselman. I'm an associate professor at the University of Toronto in the Department of Physical Therapy and also a senior scientist here at KITE. And it's my pleasure to moderate today's lecture. We're very pleased to have Dr. Field-Fote with us, who will be speaking. And I'm going to introduce Dr. Field-Fote, and then she'll give her talk. When it comes time for question and answers, just to give you a heads up, we do have a microphone that we will be circulating around. So you can definitely ask lots of questions, but just let us get the microphone to you before you start asking your question, please. So Dr. Field-Fote holds the positions of Director, Shepherd Center of Spinal Cord Injury Research Program and Hulse Laboratory, Professor at Emory University, School of Medicine, Department of Rehabilitation Medicine, and Professor of the Practice, Georgia Institute of Technology, School of Biological Sciences. With a clinical background as a physical therapist, PhD training in preclinical models of spinal cord injury, and postdoctoral training in neurophysiology, for 25 plus years of spinal cord injury research has spanned the breadth of basic and clinical translational research related to spinal cord injury. Dr. Field-Fote leads a research team dedicated to improving motor function in persons with spinal cord injury through the development of clinically accessible neuromodulation and neurorehabilitation approaches, informed by the latest neuroscience research and guided by outcomes that have meaning for persons with spinal cord injury. She's conducted randomized clinical trials with continuous funding from the NIH since 1997, with other trials in her lab funded by the Department of Defense, the National Institute on Independent Living, Disability, and Rehabilitation Research, and numerous foundations. She currently serves as chair of the Spinal Cord Injury Model Systems Project Directors Consortium, as lead PI of the NIH R25 funded program, Training in Grantsmanship and Rehabilitation Research, and as chair of the Paralyzed Veterans of America Clinical Practice Guideline Development Panel for the Guidelines Outcomes After Spinal Cord Injury. Dr. Field-Fote is a past chair of the NIH National Advisory Board on Medical Rehabilitation Research, past president of the Foundation for Physical Therapy Research, and the 54th Macmillan Lecture Award honoree. So it's my pleasure to welcome Dr. Field-Fote.
Thank you so much for that great introduction. It's really great to be here among so many colleagues that I've known for so many years, and looking forward to hearing the questions that you may have and ways that we can translate some of the work that we've been doing in my lab to the clinical setting. So I'm really honored to be invited to give a lecture as part of the Marc Rochon Distinguished Lecture Series, and I looked up a little bit about what he had been involved with, and I understand that he was very involved in really transitioning Ontario's healthcare system. And they actually wrote a book about this with his colleagues. I did not read the entire book, I confess, but I did see a foreword about it, and they mentioned that the closing lines of that book were, "Let's hope we can pick up the pace." And I think that's a really kind of fitting statement for translating research into clinical practice, because it does seem like it takes such a long time. In my lab, we're really focused on things that we can hope can really be moved into the clinical setting, and I know that that's true here with the work that many of you are involved in, and so we have that common bond about really wanting to push our work into the clinic. So when we think about stimulation and activation of the nervous system, and so I really was thinking a lot about how I should title this talk. And augmenting activation is really what we want to do. We activate the nervous system whenever we do a volitional movement. And so things that we do to add to that activation are intended to really push the nervous system to the next level to make it better at doing the things that we are trying to do with our volitional effort. And when we think about what happens when we can stimulate at all levels of the neuraxis with the different types of tools that we have available to us in rehabilitation. We can stimulate the level of the brain and cortex, at the spinal cord, and at the peripheral nerve level. And today I'll be talking about each of these and how they contribute to the activation of the nervous system in a way that's intended to improve volitional function. So when we think about one particular type of stimulation, which is what I will be focusing on a lot today, we're talking about somatosensory afferent stimulation. And that really is not the type of stimulation that we typically think of or often think of when we think of rehab. For example, excuse me, when we think of functional electrical stimulation, we're really trying to activate the muscles. When we're talking about somatosensory stimulation, we're not focused specifically on individual muscle activation. We're focusing on changing the excitability of the nervous system. And so we'll be talking a lot about different ways that we can apply that type of technology to be able to improve volitional effort. And so from the perspective of activation of bottom up, we know that sensory input is really important for tuning the excitability of the spinal circuits. We know that it also is important for activating supraspinal centers. We'll talk about why that is relevant for hand function. And from the other direction, when we talk about top down, we know that sensory input activates the sensory cortex, which is critical for activating the motor cortex. Sensory input is sensory cortical excitability is one of the primary drivers of motor cortical excitability. And so we also know that sensory input is important for guiding motor output, and it has important influences on spinal motor networks. So we're going to start by talking kind of with a condition that we sometimes don't think of very much when we think of individuals who have spinal cord injury. At least when you talk to someone who doesn't know about spinal cord injury or doesn't know someone with spinal cord injury, they rarely think about spasticity and the impact of spasticity on daily life and functioning. So what do people with spinal cord injury say about their spasticity? We're very curious about the individual lived experience of spasticity, and we did a project with our colleagues in the spinal cord injury model systems. And as part of this study, we were able to get information from over 1,000 people with spinal cord injury, and we asked them various things about their experience of spasticity. And I'm just going to touch on a couple of those points here. And that's probably true about most of what I'll talk about today. I'm not going to go into any one study in great detail. We're going to touch on the different topics that my lab is interested in, and hopefully that more additional details will come out when we talk about in the question and answer component. So one of the things that we asked people with spinal cord injury about their spasticity was, what are the things that they found most valuable for managing their spasticity? Before I get to this, interestingly enough, there were a whole host of things that people described as impacting their spasticity in a negative way. And in therapy, we often tell people with spinal cord injury and other types of neurologic conditions where there is spasticity, that spasticity can have benefits. But when people are reporting it, very few people reported beneficial effects. Less than 10% reported there were beneficial effects in 4 areas, and they were things like dressing, moving around in bed, and doing transfers. But on the whole, spasticity was perceived as something that had a negative influence on their life and functioning. When we asked them about things that were helpful for managing spasticity across the different types of injury severity, we saw that far and away, the thing that people reported most effective was stretching. So think about what's going on with stretching. Certainly, you're changing the soft tissue extensibility, but you're also very strongly activating the afferents. We know, everyone who stretches know that stretching really activates our sensory system. They also reported that things like exercise and other activities were valuable for managing their spasticity. I thought it was interesting that on the whole, exercise and stretching were thought to be more effective than medications. And that was for the people who took medications. There are many people who just say, I don't want to take medications, I'll live with the spasticity. And so the things that we have in our rehabilitation toolbox for helping manage spasticity are perceived as being very valuable. And so what does this mean for other management approaches for spasticity? And so just kind of a quick review here. So this is a very simplistic view of the very complex neural circuits that we have in our spinal cord. One thing that I'll point out here is that these 1A afferents that sense muscle stretch are very strong drivers of activation. They have direct excitatory activations with the motor neurons, and they also have a bunch of inhibitory connections to inhibitory networks that change excitability of the motor neuron. So they're very, very powerful sources of influence on spinal cord excitability. And so when we talk about things like transcutaneous spinal stimulation, you will see this in the literature called transcutaneous spinal cord stimulation. And we know from modeling studies that really it's not the cord that's being stimulated. The thing that's primarily being stimulated, are these primary afferents here that are closest to where the electrodes are and also have the lowest threshold for activation. So it's spinal stimulation in the sense that we're stimulating over the spinal vertebra, but it's not really spinal cord stimulation, which means it's accessible to many of our clinically available stimulation approaches. So transcutaneous spinal stimulation really activates these afferents, and another reason is that the dura is really non-conductive. There's been beautiful modeling studies that have been done by these groups that I've referenced here, where they've demonstrated that the structures that are activated by epidural stimulation, even electrodes placed invasively over the dura, even those primarily activate those 1A afferents because the dura is so non-conductive. And so a lot of the studies I'll talk about today touch on this idea of transcutaneous spinal stimulation. One of the ones I think is most interesting is which was a single session study that we did in individuals with spinal cord injury, where we tested different types of ways of activating those sensory fibers with things that we have available to us in physical and occupational therapy. And so the things that we tested were here in red, stretching, in green, cyclic passive movement, where the individuals in this case, where they put in a Locomat device and they just walked passively on the device. But you could do the same with a motorized cycle, for example, just to move the legs passively. Transcutaneous spinal stimulation, which is here in purple. Transcranial direct current stimulation, which is cortical stimulation, which there's some evidence that transcranial magnetic stimulation might have an effect on spasticity. So we're interested in seeing, if this more clinically accessible approach might have influenced spasticity. And then finally, sham stimulation here in orange. So let me kind of describe the sham approach. So these are all directed at assessing the influence of these different interventions on spasticity. And we measured spasticity with the pendulum test, which is a test where you extend the leg over the edge of the mat and then you watch it drop. In a non-disabled person or someone with low spasticity, that's what you'd see. It would just swing. But in a person with spasticity, you'll see it catch and bounce because the quadriceps muscle is stretched and it contracts in a hyperexcitable way. So in the sham condition here, individuals were laying on a mat for the full duration of the stimulation period. They were told that they were getting stimulation, but the stimulator was actually turned off. And so you can see that the negative value of this first swing excursion indicates that the spasticity actually got worse. So if I have a muscle that reacts in this way, that long change in angle indicates low spasticity, whereas a short or small change in angle indicates high spasticity. So you can see that it actually went in the wrong direction. The spasticity got worse. And so when we think about when people with spinal cord injury experience the highest spasticity, what do they usually say? What do they tell you about when they have the highest spasticities? first thing in the morning, right? When there's been a long period of inactivity laying around. And as we saw from the responses of people who were asked about their spasticity, activity is the thing that makes it better. And so periods of inactivity really can make spasticity worse. So here we have early effects of transcutaneous spinal stim and late effects, which is 45 minutes later. I'm going to focus on the late effects because that's what we really want, things that last for a little while. And so going back to the things that we found to be most effective, we found that the continuous passive movement, just that activity, moving the legs, and the transcutaneous spinal stimulation were the things that were significantly better than the sham intervention. A key piece, though, is this position that the individual was in is exactly the same position that they were in when they were receiving the transcutaneous spinal stim. So this transcutaneous spinal stim had to not only have an effect, it had to overcome the negative effect of just laying down with your legs straight for the whole period of the intervention. And so that suggests that this is a very powerful way that we have available to us to improve spasticity in people for whom that's a problem. One of the things that we don't know is what is the best place to stimulate. And so that's one of the studies that we've been doing recently. And this is a work that's in progress. It's been submitted and it's in the process of being reviewed, where we looked at different electrode setups to be able to see what was the electrode positioning. So this is the cathode in black, and then the anode in red. This is para-umbilically. This is over the iliac crests. We had 2 paraspinal electrodes. We had an anode, cathode, and an anode, both paraspinally. And what we found is that this setup, which has the cathode over the lower thoracic region and the anodes either para-umbilically or on the iliac crests, was most effective for actually reducing spasticity. And that's what all these error bars are here. So that seemed to be the intervention that really made the biggest difference in terms of reducing spasticity. Interestingly enough, you might think that having 2 electrodes over the spinal cord would be the most effective, and what we found is it actually seems to be interference between the 2 electrodes, and so that was the less effective approach. And these were different montages that are used in the literature, but no one's really actually compared them to see which one seems to be most effective. So here are just some outcomes from this type of testing. And I want to preface this by saying this is one of the most dramatic ones that we've seen. See if I can get the video to play. Okay, so here's one, and it's going to be really short because the file was super big. But you can see this individual has very dramatic spasticity, and then following the intervention, you see a much significant change in terms of the amount of spasticity in the quadriceps. And so let's kind of transition here to go into uses of stimulation to activate the nervous system for locomotor training. But before that, are there any questions specifically related to spasticity? Yes.
Oh, thank you. I enjoyed your slides. I work at the spastic clinic at Toronto Rehab. I just want to know why you didn't use Tardieu or Ashworth instead of the pendulum test.
Scale or the Ashworth scale. So very often when you see studies of spasticity and they use those type of metrics, because they are very gross measures, those very gross ordinal measures, it's not very sensitive to being able to pick up change. And so that's the reason that we choose to use the pendulum test. And it's actually been used in studies of drug trials as well, to be able to measure changes that might not otherwise be apparent. Yes. I'll speak loud.
Is how long after you've done the stimulation, does that effect last?
Oh, thank you so much for that question, because that's a very important question. So we measured out to 45 minutes, and we saw that the cyclic passive motion and the transcutaneous spinal stim were effective out to those time points, but it's likely that they would not last more than a few hours. But, when you think about medications, people take medications, they don't take them once a day. They take them 2 or 3 times a day. And you may know that I've done a number of studies on whole body vibration and the effects of spasticity. It's hard to carry around a whole body vibration plate with you and do it a few times a day. But transcutaneous people wears TENS units, right? And so they could theoretically use it and turn it on as they feel like their spasticity is getting worse. And so thanks for the question. I think it probably doesn't last very long. Maybe continuous use causes the effects to last longer, but even if not, then theoretically someone could use a device and use it a few times a day when they need it. Okay, moving on to locomotor training. Does training plus activation of spinal circuits improve walking function? So one thing I wanted to point out that here, the training is really the key. Whenever we're using stimulation to be able to augment function in a way that changes a person's ability to do a volitional movement, training is super important. It's the training that actually kind of tells the stimulation what it should be doing. When you think about cooking a pot of soup, right? You turn on all the burners, but you put the pot on one burner, and that's the burner that really has an effect. So it's the same kind of thing with spinal circuits. You may use a stim to turn everything on, but it's the training that actually tells the nervous system what it should be focusing on. And so the training is really an important component, and it should not be an afterthought. So here's a series of studies that we did, which were done actually in the clinical setting, looking at the effect of adding transcutaneous spinal stimulation onto locomotor training for people with spinal cord injury. And here, this little cartoon is just intended to point out that, again, this stimulation, the transcutaneous spinal stim, has its effects at the dorsal roots. And so individuals who their therapists had indicated locomotor training was appropriate for them were asked if they wanted to participate in this study. And then the therapist did whatever intervention they felt was most appropriate for locomotor training for those individuals. And so the study setup was a wash-in study. So everybody got 2 weeks of just locomotor training with no stimulation, because we know that anything that you do, if you haven't been doing anything in a while, anything you do is going to improve your functioning. And we really wanted to look at the effects of stimulation. So in the wash-in period, everybody got the same thing. And so I'm going to just kind of walk you through these. This is change in walking speed, and this is change in walking distance. The black is individuals who are in the sham stimulation group. The blue is individuals who were in the transcutaneous spinal stimulation group. The W is the wash-in period for both groups. They both got just locomotor training, and not surprisingly, both groups had a significant improvement in their walking speed during that time. However, during the next 2-week period, where this group just got sham stimulation and this group got real stimulation, only the real stimulation group actually continued to have an improvement in their walking speed. And in terms of walking distance, there's a large variability of change within each of the groups. And it was only in the second 2-week period in the group that got transcutaneous spinal stimulation that we actually saw a statistically significant change in their walking distance. And so that seems like preliminary evidence that might suggest there's value in adding transcutaneous spinal stimulation onto a locomotor training program. And so many interventions that we do in our therapy gyms are really directed at improving locomotor function, whether they're treadmill walking, walking over grounds. Some people advocate for steppers to improve walking function. There's many different things that we can do. And they essentially all build on this idea of task-specific training and activity-based therapy. The issue is that the things that are problematic for different people vary from one person to another. And so there are certain limitations that locomotor training has. One of the things that we know is that very often when doing locomotor training, the individual's in an environment where they can't make errors. We know that making and correcting errors is really important for being able to improve skilled behaviors. Also, there can be a low intensity of actual effort that the individual puts into that walking training. There's limited specificity in terms of what the individual actually needs to work on to improve their walking function. For some people, it's dorsiflexion. For other people, it might be insufficient hip extension. So there's limited specificity in terms of the locomotor training programs because everyone's kind of doing the same thing. And then there's a high cost. One of the issues that we've seen at Shepherd Center, where we have a very intensive locomotor training therapy program and also a post-rehabilitation training program, is that people often come in and they participate in these intensive programs and they go home and they can't keep up the practice and they don't keep up the skill that they developed. And trying to keep up that skill is very difficult because it's costly and it requires a professional to be able to work with you. So those are things that drove us to actually think, are there other things that we might be able to do to overcome these limitations in our locomotor training programs for people with spinal cord injury? And can we make that an accessible intervention that theoretically could be rolled out into the clinic, into the community wellness setting, and maybe even into the home if someone has adequate safeties for balance. And so, excuse me, this is the circuit that we have been working on developing, and this is the work of Nick Evans, who was an exercise specialist, which I understand here is called a kinesiologist. And it involves tasks that are really, they are not locomotor training, but they're specific tasks that require skill and are components of walking. And so being able to do lunges in different directions, being able to get your foot up off the ground in a step, wall squats to develop strength, jumps to be able to develop explosive force, hip hikes and marching in place very quickly, and then a rest period that includes dorsiflexion. And so these are the components of this circuit that we believe that contribute to it being not task-specific, but task-related to locomotor training. It's accessible, it has cyclic movements that are done full weight bearing, they're done at high velocity, and they're physiologically intensive. And so we were informed by the literature that suggests that even moderate intensity activity, like 50% of heart rate and higher, has an effect on brain-derived neurotrophic factor in the system that seems to drive skill development. And so these are all done at an intensity to keep the heart rate up to at least a moderate level of intensity. And if we have time at the end, I'd love for someone to ask me where this idea came from, because it's kind of an interesting story for those of you who know Janet Carr and Roberta Shepherd. And I'd love to tell you, but I'm not sure I'll have time to go into it as part of the talk. So here is Nick working with an individual with spinal cord injury, providing assistance when needed, but mostly providing support for balance. And here are the outcomes that we observed from that trial. So the things that we're primarily interested in were walking speed and distance. So let me back up for a second. This was part of Nick's PhD thesis. And in that thesis, we were interested in knowing not only about the effect of the motor skill training, but also whether augmenting that training with brain stimulation, transcranial direct current stimulation, made a difference in the outcomes. Because we know that those descending drive to those spinal circuits is important for activating the spinal circuits. And so essentially the bottom line of that story is that there was no difference in the brainstem group versus the no brainstem group. But across both groups that we saw an improvement in walking speed and improvement in walking distance in both groups. In addition, and this is a slide I just put in for Kristen and her group's interest. Sorry, I keep hitting you with the laser pointer, don't I, Kathy? Sorry about that. So we looked at the Berg balance scale and the Falls efficacy scale, and we saw that collapsed across groups, people had improvements both in their measured balance performance and in their feelings of being safe and being able to walk without falling. And so here's kind of just to make the point. Here you see people with different walking function, despite the fact that they all have the same level of spinal cord injury. And so the idea is that that motor skill training program might be able to be customized in a way that, you know, someone focuses on dorsiflexion, if that's the piece that they need to concentrate on. Or if they can't develop explosive forces, they focus on jumping. I had a gentleman call me. His physical therapist was going on maternity leave, and he had read this article, and he asked, you know, could I do this at home while she's on maternity leave? And I said, yes, you know, as long as you have someone in your home that you feel can be there and make sure you're safe. And so he did it, and he said when she came back from maternity leave, she was amazed at what he could do. He said, when I first started, I couldn't get off the floor, but then I was really jumping, and I'm like, I wonder if you really were, but at least it felt like you were getting up off the floor. And they said it really made a difference for his walking function, and he was able to actually do it. It was feasible for him to do it in his home. We're going to shift gears now and start talking about hand training. And so before I leave that, are there any specific questions about locomotor training before we kind of shift gears? Suk, I thought you'd save your questions for the hand.
I will. I'll ask more. This is actually probably more of a collective. So when you do locomotor stimulation, where exactly do you apply the stimulation, and what are you stimulating? I know
Yeah.
you mentioned TDCS, but are you also doing spinal stimulation?
Yes, so in that TDCS study, we were just doing transcranial direct current stimulation. But we are now our studies are looking at combining the motor skill training with transcutaneous spinal stimulation. And we are putting the electrodes around T11, T12, and the idea is that they're activating the circuits that are descending in that area, which are mostly the quadriceps that are being targeted in that area. And that's a great question because, you know, maybe in some people we want to put it further down and target the soleus. But right now we're just focusing on quads. Cathode. The cathode is over T11, T12, and then the anodes are either para-umbilically or on the iliac crest.
Hardware. Have you done this with hardware?
Oh, yes. You mean what type of stimulator?
No.
Sorry. Yes.
Surgical.
Yes, thank you for that. That's a great question, and it's really relevant to the part that's coming up in hand function, because that was a big question we had in this onward trial that I'll talk about. And most, today, the hardware that's implanted is not conductive. It's titanium. If someone came in with older hardware, we'd probably have to go back and try and check. But so it's not conductive, so it's not a safety issue. But I think there probably are questions about whether you really are getting at what you're trying to target. But you know, the nerve roots extend out over, and so it's possible that we are.
You mentioned Carl and Shepard. I use their approach all the time, motor relearning. I use that as a theory, like overarching. Do you do the same?
Well, we focus. When I think of Carr and Shepard, they haven't published a lot of research. Most of their work has been like clinical application, but I think of them as being the moms of task-specific training, and so that's what I mean. And I actually gave a talk and shared a platform with Roberta Shepard, which was kind of where this idea came from, and it's a kind of funny story, and I'll hope I have time to tell you it.
In regards to, I guess, this concept of influencing the general excitability of the spinal cord and priming it, I guess, for training, what are your thoughts about combination therapy with drugs combined with spinal stimulation?
Yeah, that's a great question. I mean, when I think of the drugs that have been tried in spinal cord injury, I mean, certainly that's an area where there's always progress and new developments, but at least the things that I've seen published, I haven't seen anything that I thought was so fascinating. We thought, oh, we have to try this in combination. If you have other thoughts, I'd be interested in chatting with you about those.
While, but I was thinking of their studies like Barbeau, which has been
Yeah.
quite some time.
Yeah, but those, I mean, the big drawback about, I think of like clonidine, which is probably the big one, such terrible side effects. I mean, you have to have a life too. So, I mean, that's a big drawback of things. Even if they potentially are effective, the side effects can be really problematic. There's one more question over here.
I'll try and be quick. The question's sort of two parts though, and that is, are you doing any electromyography at the same time to see the muscles? And the reason I'm asking the question is I'm thinking about the motor cortical reorganization that happens for the different muscles, and it often has specificity.
Yeah, so that's a great question. It's about whether we're doing EMG to assess changes in EMG. I had a super smart engineer in my lab 3 years working on EMG and trying to figure out what it meant, the changes that we saw with our EMG. And it took 3 years, 100% of our time devoted to that, and we found nothing useful. And so I have not been focusing on that at all. But thank you for the question. So I'm going to move on now to hand function. And the question is, does activating cortical circuits directly or indirectly best augment hand function? And so we have used different approaches. This is some older work that we did, but I think it's very informative, showing that with just peripheral nerve stimulation, like TENS, we activate the nervous system in a way that can actually help reorganize the brain to make it more functional for movement. So kind of to orient you here, this is a transcranial magnetic stimulation map, a map that's constructed. Here's CZ, which is the vertex, and we would expect in non-disabled people that the primary activation, if you're talking about hand function, would be coming from forward of CZ. And you actually see that there's disorganization where, in fact, the sensory cortex seems to be making a much more than usual contribution to the upper extremity motor evoked potential. But after training, you see there's a change in the cortical map so that more of the information comes from anterior, where you would expect it to come from. So these interventions don't just change function, they change the nervous system. And the question is, like, which one comes first? And we don't really know, but they seem to change together. And so we did some studies looking at the effect of different types of stimulation, either peripheral nerve stimulation or vibration or brain stimulation, to see which one was most effective for improving hand function in people who had tetraplegia. And so here, I'm just kind of separating these out. We have a group that got peripheral nerve stimulation, which is just TENS applied at the median nerve, vibration, which was applied over the tendon to activate those same kind of afferent circuits, and transcranial direct current stimulation. And what you see here for pinch force, 9-hole peg test, and this one, which is a fine motor tracking task where they had to increase and decrease their pinch force in order to follow a sinusoidal wave. The intervention that had the largest effect was peripheral nerve stimulation combined with training, and we're looking at effect sizes here across over the 0.5 bar, and the asterisks indicate statistical significance. So the largest single effect came from peripheral nerve stimulation combined with this training. We also saw effects from transcranial direct current stimulation on all the different measures. And so either of those approaches, which can be clinically accessible, seem to be valuable as part of a program to improve hand function. And so here you see the things that were most dramatically affected. We recently completed a pragmatic clinical trial where we tried these approaches in people with early after spinal cord injury, where we have such great variability, and we're still kind of slogging through the data at this point. However, we've seen some impressive changes in motor evoked potentials, before and after training in the thenar muscles. So it really does seem like it has an effect at that level of the nervous system and on cortical activation. So how are these findings relevant to spinal stimulation? And now I'm going to be talking about a study that we did that was recently published and is part of the device development for a device that is not yet FDA approved. So these next slides that I'm going to show you, which have a blue background, are from a device that's not a clinically accessible device, not yet FDA approved. And this was the Onward study. And so in this study, we devised a motor training approach that focused on the different tasks that could be done with the hand, and it was combined with transcutaneous spinal stimulation. I know that Suk, you may know, was an important site leader in this study as well. And so, sorry, I'm laser pointing at somebody again. Here's kind of just an overview of the study design. It was a wash-in trial where people who were enrolled got 2 months of just rehabilitation and then 2 months of rehabilitation plus stimulation. And we had assessments at baseline, at 2 months, and then at 4 months. There were 65 people who initially were enrolled. 64 were included and began the study, and then 60 actually completed the study. And so here are the primary endpoints of the study. So in order to be considered a responder, the individual had to have a change in strength, in one of these measures of strength, and a change in one of these measures of function. And so that's a relatively high bar to say someone has to have changes in 2 different metrics in order to be able to be considered a responder. But because this is an early study, we were also mostly focused on the safety. And so at the end of the study, the analysis demonstrated that 72% of the individuals who participated were in the responder group. So they had at least a change in at least one measure of strength and at least one measure of function. And here are kind of the outcomes across the different metrics that were used. I honestly think the things that were most impressive were in the gray bar, which you can hardly see, sorry about that, are the training only periods. And then in the red line is the training plus stimulation. And so the things that I thought were most impressive were actually the changes in strength. And so you see here, there's hardly any change in strength prior to, or at least it plateaus, prior to the onset of the stimulation. And then it really takes off again after the stimulation. Sensation was not one of the categories of whether or not someone responded, but there were also impressive changes in terms of sensation. And when you think about that, there's evidence from the sensory stimulation literature that activates the sensory cortex. And so it's probably not surprising that this powerful stimulation that activates afferents had an effect on sensation. And so the trend suggests that further improvements may be possible with continued stimulation, because you see in many cases, the trend lines are continuing to go up at the end of the training and intervention period. So this is probably the piece that's most interesting, is who seems to respond. So here's our functional responders, our strength responders, and then the overall responders. And you can see them parsed out by individuals with AIS-B, C, and D. And far and away, the group with AIS-C was the group that got the largest changes in function. Individuals with C and D had the largest changes in strength. And surprisingly, in some ways, individuals with AIS-C had some responses as well, both in terms of strength and in terms of functional outcomes. So what I'm interested in doing in our secondary analyses is going back and helping try to figure out what is it about the people with AIS-B who had the responses that we might be able to tweak future interventions to be able to make the stimulation more valuable for those individuals. So some of you may know this guy. He's quite a character, Alan Brown. He's had spinal cord injury for over 30 years. And he is currently the director of new partner engagement for the Nielsen Foundation, the Christopher and Dana Reeve Paralysis Foundation. And so I've known him since very early in my career. And he saw me at the Asia meeting. And he said, "Hey, Eddie, look what I can do now." And he gave me a cheers. And he had not been able to lift his own beer prior to the training. And it really made a difference for him, even though he's many, many years post-injury. So I'm going to wrap up by talking about a systematic review that was done by a physical therapist who was in my lab for her PhD and then went on to do her postdoc and did this study as part of her postdoctoral training, where she looked at all the literature related to experimental interventions for people with spinal cord injury across drug trials, across biologic trials, device trials, and essentially what she found is for acute and chronic spinal cord injury, far and away the highest evidence was for phase 3 trials where they had a component that included rehabilitation. So rehabilitation is really an essential part of whatever intervention is tried experimentally or clinically for people with spinal cord injury. And so kind of just to summarize, afferent input in all its forms can modulate spinal excitability and cortical excitability. Clinically accessible stimulation can be a powerful adjunct to training. And we have to consider the specificity of training beyond just the kinematics of the training. So more than just repeatedly walking, and thinking about what are the specific things that this individual needs to improve their locomotor function. And I'd like to thank my funding sources that contributed to our success and my amazing lab team. And I hope we have a chance to see you all in other places sometime soon. Thanks, and I'm happy to take questions.
Hi, thank you. I truly appreciate your talk. First, a comment. I think electrical stimulation, especially, has been seen for far too long as a on and off function. Let's put whatever it is, if it is transcutaneous, if it is over the muscle for functional stimulation, turn it on, activate as much as you can, and get as much out of it as you can. And the specificity of training and thinking about resistance, thinking about strength, was fully forgotten forever. So I truly appreciate that you are taking the specificity of training with electrical stimulation in mind and with transcutaneous stimulation, transcutaneous spinal stimulation in mind. So that's awesome. Appreciate that. For the lower limb stimulation, I think you didn't talk too much about what stimulator have you been using, what sort of electrode frequencies and so on.
Yes.
Because there is also too much literature on things that no one will ever get access to. Yep. While everyone in the clinic here have a $300 stimulator that probably can get the job done. So what have you been using and what are your thoughts on that?
Thank you so much for that question. You know, I totally agree with you. There are things that make the $2,500 stimulators different are probably not things that are tremendously valuable, or if they are valuable, they're not that much more valuable. So things like a kilohertz frequency, things like the 1 millisecond pulse duration. I had this great slide from Panzini, which is like 1976, where he showed this input-output curve where what's the threshold when you use a longer versus a shorter pulse duration. And yes, the threshold is lower when you use a longer pulse duration, but just by turning up the intensity, you can also get that same level of activation. And so yes, I agree that the stimulators that we have in the clinic can do the job. And the pragmatic study that I talked about where we use it as part of the clinical intervention for locomotor training was an EMPI continuum device. And we used in that study 50 hertz. In hindsight, we should have used 30 hertz, but we did it when the literature was very early. Literature is now suggesting that lower frequencies like 30 hertz seem to preferentially activate those motor circuits, whereas higher frequencies seem to activate those inhibitory interneuronal circuits. And in the studies that we're doing now, we're looking at 50 hertz versus 80 hertz. And we're actually seeing some really cool things with 80 hertz. So thanks for that question, very important. You get the prize.
Super quick question. When it comes to the locomotive training, did you notice there was a difference between if you were doing overground walking versus treadmill training or even body weight support treadmill?
Oh, thank you for that question. I will refer you to our 2011 article in Physical Therapy, where we actually tried to answer that question. We compared locomat training to overground training with body weight support, but not with body weight support, just having the harness on for safety, and common perineal nerve stimulators to get some dorsiflexion versus treadmill-based training where the therapists had their hands on and were helping move the leg, to treadmill-based training where they used common perineal nerve stimulation to trigger a flexion withdrawal response. And I was sure that the flexion withdrawal response was going to be it. And I did that study because I had a colleague who was sure that the hands-on training was going to be it, and we were both wrong. It was the overground training. And I think it's because, you know, treadmill training, you can go faster, but it's not, when you think about, like, how many of you have run on a treadmill? You know, it's not the same as walking over ground, as running on the ground. Something's different. And I have a colleague who's a biomechanist. He says, no, it's the same. The ground reaction force is the same. Kinematics the same. Like, your nervous system knows it's not the same. So whatever you're learning on the treadmill is not 100% what you need to know to walk over ground. And I think that the environment is really important.
You used the word specificity, and the general trend that's going on, at least I think in the United States and sort of here in Canada, is precision medicine, which actually has a component of specificity. Yeah. What do you think about the fact that we need precision physical therapy in the context of all of the things that you're showing, and how do you think we can get that message across that we need to do that if we're going to get the answers, the best answers we can get?
That's a great question. So I think traditionally when we think of specificity, we think of, you know, if you want to improve walking function, you don't do cycling. And you work on walking and you do locomotor training. But I'm thinking along the lines of what you just said, that we need to be more precise. A person doesn't just need to do this all day. It's not going to make him better at doing this. If his problem is that he can't get his foot up off the ground, then we need to work on the dorsiflexion. If the problem is that he's not getting enough hip extension, which is so powerful to drive those circuits in the spinal cord, then that's something that needs to be trained. And so I think that those two things need to go together, that when we think about specificity, we need to think about specificity in terms of what does this specific individual need to improve that function that they're trying to improve on.
Yep, okay. I have one question more about spasticity with respect to the upper extremity. So we have seen now in the literature some of the work done around using a TENS unit in the thoracolumbar region to reduce lower limb spasticity. Have you guys done it with the upper extremity in the cervical spine? Or has anybody, for that matter? I don't think I've seen it.
But as far as, thanks for the question. So so Jim Guest, who was one of the sites in the Onward study, he actually played around with the stimulation parameters to see if, because I think he had 2 people there, and he had one of them for whom spasticity was a real limitation. And he did some playing around with the stimulation frequencies, and he says that he saw some decreases in spasticity. But, I mean, one of the big problems is measuring spasticity in the upper extremity. I mean, it's hard enough in the lower extremity. It's really problematic in the upper extremity. So I think it makes sense that there would be value in the uppers as well.
Hi, great talk. I believe you used to work on FES for locomotor training. using
So
FES?
we
and then
use FES, so from the perspective of we use stimulation as part of a functional task, but it wasn't activating muscles the way we typically think of. We used it to activate a common perineal stimulation to elicit flexion withdrawal for dorsiflexion, knee flexion, and hip flexion at the same time. So that was the application of lower extremity FES that we used.
my question was, I thought that you used to use FES for the intervention, but now you use TSS mainly. I wonder your thought about the comparison between the two technologies.
Yeah, so the question is comparing FES to transcutaneous spinal stimulation, and that's a very fair question. So I have not used FES in the way that you have timed activation of the lower extremity. The only study that I did where I used stimulation as part of locomotor training was to trigger a withdrawal response. So 1 point of stimulation to get this as part of walking. And in that group, they were walking on the treadmill. So the treadmill brings the leg back, you trigger the stim, it comes down in the right place for the next step. I think one of the limitations that in the real world of the clinic, at least in the US, FES for walking, where you have to time the activation of the lower extremity muscles, the limitation is equipment. Transcutaneous spinal stim doesn't have, it's not as powerful, but it likely recruits in the same order that you think of when you think of physiologic recruitment, so that there's probably less likelihood of fatigue. But I understand there's some really interesting work going on here where people are looking at electrodes that are intended to overcome that issue. So that seems like it would be really helpful. I look forward to talking to you about it.
Thank you.
First of all, fabulous talk and also fabulous responses. You made the comment about the walking and aspects, and a few minutes ago when we were talking about on different approaches to walking and the specificity of that. Walking also takes balance, and if we think back to the original work in balance, vision has such an important part. Have you done anything with blind walking? Because if you actually look at people who are blind, they walk very differently than people not. And have you ever had any subjects who have also, been blind and spinal cord injured? That would be to get an idea of how much the other sensory inputs have an effect.
That's a really interesting question. I've never had a participant who was vision impaired, but I can imagine, like we see in other types of training, where if you have one sensory function that is lower, that the others kind of do more work. So maybe that would increase the reliance on somatosensory input, and you would actually get a bigger bang for your buck. That's a really interesting thought. So there's a research question if anyone is looking for something to investigate.
So I think in the interest of time, if it's okay, we'll wrap up now. For the research trainees in the room, there is a lunch that'll be happening in B10, so feel free to head over there afterwards, and Dr. Field-Fote will be there, and you can have a chat with her. But thank you so much for a really wonderful talk, lots of discussion, great questions, everyone. We'll just give you one last round of applause.